Welcome to the GSA Annual Scientific Meeting Session, Latest Perspectives and Future Directions, Effects of Loneliness and Social Isolation on Cognitive Health. We have few housekeeping things to go through, but first, please introduce yourself in the chat. We would love to know who's here. We also will use that chat box throughout the program, so please comment or ask questions. Our program is being recorded today and will be archived on the GSA website. We ask you to keep your video off and audio on mute until it's time for discussions. A question and answer session will be immediately follow the live presentation. My name is Paras. I'm a physical therapist working in San Jose, California, and my co-moderator, Amanda Sonnega. She is an associate research scientist at the University of Michigan. Today's iSession is part of a series planned by a GSA program publication and product committee work group to provide participants with a forum for conversations on findings, limitation, future work, and advances around an interdisciplinary topic. Without further delay, I will hand the microphone to the chairs of today's session, Dr. Ted Ng, PhD Assistant Professor, Physical Medicine and Rehab, ASU.
Thank you, Paras, for opening up the session. So let me share my screen. Let's see. Just to make sure, like we talked about just now, let's see from the beginning. And I think you are still seeing my presenter slide.
Yeah.
So are you seeing the presentation right now correctly?
It's good, right?
Okay, thank you. So yeah, as Paras introduced just now, I'm currently an assistant professor. Well, I just changed my institution to Rush University Medical Center this summer and started at the Rush Institute of Healthy Aging. I'm Ted Ng. And today we will be talking about latest perspective and also future direction concerning loneliness and isolation and how they are related to cognitive impairment as an outcome. So I will first provide a general overview, and then after that, we will talk about some of the updates to the 2021 GSA sessions, and then I will end with some pointers and future directions that we have for the field. And acknowledgement, so part of the studies that I will be showing later on has been called with one of my co-author, Dr. Yu, who I believe is also present in the audience here. And so just to have everybody on the same page, because I understand that perhaps not everybody is familiar with isolation and loneliness, although we heard it a lot on the social media and also news. So isolation refers to the more objective measures of being socially disconnected, whereas loneliness refers to the more subjective feeling of disconnection or perceived inadequacy. Whereas cognitive impairment, in my subsequent slides, it is defined differently by different investigators and different people. But for the presentation today, I define it as CIND, cognitive impairment without dementia, or MCI, mild cognitive impairment, and also dementia. So two different stages of cognitive impairment. And this relates to the social neuroscience of dementia, because social isolation and loneliness, here I can refer to SIL, they have adverse effects on a lot of health outcomes in older adults, as probably a lot of you are aware, but cognitive impairment is really one of the more prominent ones, especially in older adults. And they are theoretical propositions that actually has been proposed in the past 1 or 2 decades that these two as stresses has been evolutionary conserved throughout humans' evolutions, because when we were more hunter-gatherer kind of era, which is many years, many decades, or even centuries ago, we will have evolutionary advantage, and we will survive better without isolation and loneliness. So this theory actually postulates that with these two stressors, they will elicit downstream mediatory pathway, which results in adverse health outcome, and a range of them, which also includes cognitive impairment, which is the focus of today's iSession. However, although they are already these theoretical propositions, but they have been lacking empirical data. So primary data analysis on findings supporting these two or even more theoretical framework has not been a lot, and there has been, to my knowledge, less than 10 studies. So the presenters today will present some of those few studies that we are actually conducting or have been conducted. And of course, this very prominent lack of empirical data, which is a gap in knowledge, has also been highlighted in the consensus report that the national academies have come out with about 2 to 3 years ago. And I think this is a very great consensus report, although it has, I think, about over 100 pages, but it is definitely a very defining and also very informative report that I guess if you have interest in this field, are definitely worth a read. And of course, there have been many studies conducted on loneliness and adverse effect, and definitely loneliness and risk of dementia has been so-called validated, but there has also been contradictory findings which found that loneliness after controlling for confounders actually do not precede the risk of dementia or cognitive decline. So the jury is still out there regarding whether loneliness is definitely related to dementia or not. So the association between loneliness and biomarker has been also a field that has been gaining traction. And you can see that loneliness have been also associated with inflammatory markers, because as stresses, it can elicit the HPA axis, which is a stress reaction in human body, which can further elicit downstream pathway, and one of the prominent one is inflammatory markers. And so loneliness, isolation, and inflammation has been investigated pretty extensively, and this meta-analysis that I screenshot here actually shown that they have been differential association between these two loneliness and isolation construct with slightly different inflammatory markers. So you can see that there are a few constructs at play here, while this third screenshot at the bottom right, you can see that this is, to my knowledge, the first paper that actually examined all these constructs together in one single paper, which I think is actually a pioneering paper in the field. And it examined health factors and including biomarkers as well, which is not reflected in the title, as potential mediators of the effects of loneliness on cognitive abilities in general. So it actually integrates all these different constructs and factors together in a paper, and I will talk about, elaborate on that a little bit later on. So this is the current state of the art and also the knowledge gap that we have right now. So as you might be able to infer from what I presented just now, there is definitely an overlooked physiological link in terms of how loneliness can so-called get under the skins to affecting changes in biomarkers or associated with biomarkers, and which further leads to cognitive decline or cognitive impairment. And secondly, there's also, although not a so-called deal breaker per se, but there are a lot of studies which use continuous cognitive functioning score, MoCA, MMSE, composite neuropsychological tests. Of course, they are useful in their own aspect, but I would champion that actually cognitive status, which is more clinically relevant, because cognitive status here identified as the different cognitive stages of dementia, including subjective cognitive impairment or declines, objective cognitive impairment, including MCI, CIND, which ultimately leads to Alzheimer's and other type of related dementia. So categorizing cognitive status is also one of the way to further push the field forward. And there have been composite measures. And the first study that I highlighted just now in the previous slide actually used a composite measure, which is the allostatic load measure. So allostatic load measure actually integrate about 10 or so, depending on different investigator and data set of biomarkers into one single composite or measure or construct, and use that as a mediator in the paper that I showed you just now. So the paper actually showed that allostatic load did not mediate the so-called effects of loneliness on cognitive impairment of cognitive decline longitudinally. So in response to that, there has been at least 2 commentaries published in response to the paper that a targeted approach of individuals' biomarkers instead of a composite measure might be more appropriate to further investigate this underlying mechanism. And of course, lastly, there have been debates on potential, to me, it's very likely residual confounding effects of a lot of other factors, because isolation and loneliness are social constructs. They are important, but they are entangled with a lot of other constructs, and it is a little bit messy, if not controlling for other constructs. They are closely relevant to both the isolation, loneliness, and also cognitive impairment. So with that, my co-author, Dr. Yu, and I have conducted this study, which is recently published in JGMS and also GSA journal. We attempted to see whether 5 different inflammatory markers that are readily available in the HRS, in indicating different physiological system each, would actually mediate the longitudinal association between loneliness and incident cognitive impairment. So as you can see here, this is the conceptual model, the theoretical model that we actually constructed based on the theories that proposed, which I shared with you in the previous slides. So we integrated these different constructs together in a single model and postulate that loneliness as a stressors could elicit biomarkers in a subsequent wave, and which in turn leads to incident cognitive impairment. So this is the hypothesized biopsychosocial model of which we base our hypothesis and also aims on. And very briefly going through the result and also the discussion. So because the 2021 iSession, my co-author Dr. Yu actually shared that the prelim result. So this is the finalized and also published result. So the candidate biomarkers, as mentioned, we have 5 different candidates biomarkers, each indicating slightly different metabolic and also physiological dysregulation, as you can see down here. And we show, we actually show that after controlling for confounders, none of the longitudinal mediation model were significant, although controlling for confounders or covariates, the cross-sectional associations or mediations were still remain significant for the 5 biomarkers. So this is pretty complicated to disentangle. So this will be the next slide. So for longitudinal mediation, after controlling for covariates, we show that there is an attenuated association between loneliness, biomarkers, and cognitive impairment. So actually, this is pretty similar to some of the findings that has been shown in the literature, because a meta-analysis actually shows similar confounding effects of covariates or confounders, however you would call it, on confounding this association between loneliness and cognitive impairment, or in general, cognitive abilities or decline. So there has been consensus on perhaps social isolation would be so-called a more objective measure per se, although it is not entirely objective because it's also self-report but it could potentially be a more objective and also a more prominent and a more robust association with cognitive impairment instead of loneliness. And I think Dr. Shi actually showed that in some of his study recently as well. So perhaps also there are also alternative underpinning pathway. So perhaps we have been barking at the wrong trees. Health status maybe could be the mediators instead of biomarkers. And of course, I think Fereshteh might have shown something, Dr. Shi have shown something such as psychological well-being as mediators of this pathway as well. So instead of focusing just on biomarkers, we can focus on a lot of other mediators that could mediate or so-called get under the skin of how loneliness/isolation actually leads to cognitive impairment. And this so-called health status as mediator has been actually collaborated also by theoretical proposition, but to my knowledge, there's only been one study which has examined this mediational pathway. So a little bit of conclusion and also how these results and also taken together with the literature, we can inform future research. So there are about 5 points here that I'm providing. So we provide a biopsychosocial model that's specific to this different construct and this mediational pathway, which is theoretically informed, as I've shown, and also as we have shown, empirically examinable. So we highlighted the importance of controlling for confounders, as you probably can see by now, and especially when comparing cross-sectional and longitudinal result, because we couldn't infer the cross-sectional and longitudinal findings would be the same even using the more robust models such as SEM or CRPM, because as we've shown in our paper, that is not the case. And also, as you can see on the third point, although a lot of study has actually shown cross-sectional significant bivariate association between either loneliness, isolation, or both with biomarkers. So they have shown this bivariate association, but the third construct or wearable cognitive impairment or functioning is actually not taken into account in the model or in the theoretical framework. So that is, so what I'm saying is that the third point would establish a baseline or the so-called framework or the groundwork for further investigation, but that should not be the case going forward, because without cognitive impairment as a third construct, that wouldn't really be very informative. So lastly, we in our paper also suggested other suggestions for future investigation, which I do not have time to go through here. So additional considerations or future directions that I have not talked about in the slide nor in our paper is that a lot of you that are in epidemiology or cohort study will probably be very familiar. So reverse causality, my 2 other panelists will talk about this later on as well in their slides. So a lot of so-called contention about how social isolation and loneliness, they are so-called the more social construct, and to some more biomedical focused investigators, they will think that they are social constructs, so they shouldn't be actually predicting dementia, cognitive impairment. So sometimes as investigators in this field, we had a hard time of convincing people with our result, even showing our result that, you know, this is actually a robust association, especially with social isolation. So reverse causality and also a few other points here we should take into account when performing our studies and also when publishing our result as well. So there are some other relevant studies that I have performed in this field in my recent venture. So the first screenshot here, the JGMS here is the co-author paper that I've shown the result for. And then there are some other papers that I have churned out in the past few years, which are closely relevant to this topic. And of course, one of them I forgot to highlight is a co-author paper with the panelist, one of the panelists, Dr. Shi here as well. So this is my last slide. as I would like to kind of do a little plug here. So this is a special issue that I'm co-editing with several prominent investigators in this field of isolation and loneliness and also social network. So we are actually accepting submission right now for the abstract through 30th of November, and for the full manuscript, the 30th of April. So we already have about 10 or so manuscripts going through the review pipeline. So with that, feel free to email me with any questions that you have regarding the presentations or the special issue. So with that, I'll thank you for your attention and pass the time to the other panelists.
Thank you, Dr. Ng.
Thank you, Fereshteh. You're up next, and are you ready to share?
Yeah, thank you.
Okay, wonderful.
Hi, everyone. So I'm sharing my slides. Okay. So hi, everyone. So I'm giving an update of my last presentation, GSA presentation on the longitudinal impact of social isolation on cognitive function among frail old adults. So just a brief overview of my last GSA presentation. So the specific aim of that presentation was to examine the association between changes in social isolation and cognitive function among community dwelling old adults in Quebec, in Canada. So in order to address that goal, we used 3 waves of the frail longitudinal study in Quebec, which was conducted among 1,643 older adults between 2010 and 2012. Sample was limited to ages 65 and over. And for a statistical analysis, we use latent growth curve models, which include 2 features, intercepts and slopes. So intercepts means that we look at the association of social isolation and cognitive function at baseline. And slopes refers to the changes in social isolation and cognitive function over time. We measure social isolation by social participation, social networks, and social support from different types of social ties, such as friends, children, family members, and partner. We measure cognitive function by Montreal Cognitive Assessment, and our covariates included age, sex, education, income, and life habits, such as smoking, sleeping patterns, and alcohol consumption. So our results demonstrated that in terms of the association between changes and the slopes. So we found that social support from friends was associated with better cognitive health among old adults over 2 years. And in terms of the relationship between intercept and slopes, means baseline and longitudinal impact, we found that participation in social activities, social support from family members, and the presence of friends at baseline were protective against cognitive decline among older adults over 2 years. So what happened next? This is in continuous of that work. As Ted mentioned in his talk, there are really few research studies that look at the biological factor that maybe impact on the relationship between social isolation and cognitive function. So we look at frailty as a biological marker. We look at the moderating role of frailty on the pathway from social isolation and health outcomes. And we submitted this study to the special issue of social isolation and loneliness at BMC Public Health. Then we revised it just recently, so we hope that it published soon. So the specific aim of this new development was to examine whether the relationship between changes in social relationship and cognitive function varied based on changes in frailty. So we hypothesized that greater social relationship would lead to better cognitive function among frail older adults rather than robust older adults. So the rationale is that social isolation is a stressful condition that can lead to serious health outcomes, including cognitive impairment. And the process is similar to frailty, because frailty also can lead, physical frailty can lead to multiple adverse health outcomes, such as cognitive function, depression, falls, and so on. So frail older adults are vulnerable to life stressor due to their low level of physiological reserves compared to robust older adults with high level of physiological reserves. So we want to, we hypothesize that the impact of social isolation on cognitive function can be different among frail older adults with low level of physiological reserves compared to robust older adults with high level of physiological reserves. So our results highlighted that social relationships are linked to better cognitive function among frail older adults, but not among robust older adults over 2 years. So the underlying mechanism is that robust older adults have a high level of physiological reserves. So they can, that high level of physiological reserves can compensate for challenges related to aging, and they can cope better with health stressors, so they can recover more quickly from illness, or they can maintain their health status without receiving any social support from the others. However, social relationships can compensate for those age-related challenges among frail older adults. So they can overcome with life stressor and they can recover from illness. In addition to that, if we look at the different types of social relationships like social networks, social support, and social relationships, we found that social support rather than social networks is linked to cognitive health among frail older adults over 2 years. So it means that meaningful social relationships and the quality of relationships are more important than regular contact with relatives or close friends. And the quantity of relationships are not really important. So the meaningful social connectedness can have more health benefits among frail older adults. So again, the assumption is that social ties may, supportive social ties may reduce the impact of stress and improve physiological responses to life stressor among frail older adults. So consequently, they can improve their health status. So social support can compensate for physiological stress and for frail older adults, so they can recover more easily from illness because of that feature. So our study we conducted in a really short period of time, just 2 years. So it's really important to replicate these results with a longer period and larger sample size, like national data set like HRS or ELSA data set. And also we couldn't rule out the reverse causality in our study, but currently, for example, now it's important to look at the bidirectional or the causal relationship between social isolation or loneliness and cognitive function. And also the bidirectional association between social isolation and frailty. So currently we are looking at the bidirectional association between social isolation and loneliness with physical frailty. Using LESO dataset from the Netherlands, we use the 7 waves. So it's really important to do this also with social isolation and cognitive function. So we need to understand whether social isolation can lead to cognitive decline over time, or cognitive decline can lead to social isolation or loneliness over time. So it's important to use like statistical analysis, like random intercept cross-like panel models to better understand the relationship between these variables. And also we focus only frailty as a biological factor. So it's really important to consider other health risk factors that maybe impact the relationship between social isolation and cognitive function, such as sedentary behaviors or physical activity. And it's also important look at social isolation and loneliness together, because in our study, we look at social isolation like we combine social participation, social network, and social support. But it's important to look at objective social isolation and loneliness. So we look at quantity and quality of relationship together and how they can impact on cognitive function over time. In addition to that, it's important to understand whether racial, ethnic, and gender disparities can have an impact on the relationship between social isolation and cognitive function over time. So thank you for your listening. I pass over to the next presenter.
Can you see my slides okay? Okay, great presentation, Fereshteh. Yeah, okay, both of the presenters pointed out the reverse causality issue. So my topic is quite different, that use the causal mediating analysis trying to rule out the issue on bidirectional association. Okay, thanks for having me. This is Xiang. I'm a research scientist at New York University, Dory Miles College of Nursing. And my topic today is on social disconnection, accelerated biological aging, and the risk of motor cognitive risk syndrome. As mentioned before, social isolation and loneliness are significant public health threats that increase the risk of a lot of health outcomes. And both of the conditions are prevalent. It's estimated that 24% of older adults are socially isolated and 43% of older adults in the United States feels lonely. But there are two different constructs. Loneliness is defined as the subjective feeling of isolated, but social isolation is the objective state of being isolated. It is defined as a lower level of social contact. And the loneliness is an unpleasant feeling, but the social isolation need not be unpleasant. It may be chosen. Why I say they are both public health pandemic, because from the data drawn from the Pew Research Center, we can see that the average household size has significantly decreased, and the number of people who are living alone and the number of single occupancy household have been greater than ever in recorded human history. And we are seeing there is an increased rate of childlessness and a decreased community involvement. And the percentage of Americans reporting no religious affiliations has increased a lot over the past decade. So why social isolation and loneliness has led to so many health outcomes? There are now three potential pathways that could explain the association. The first is health risk behavioral pathway, which means older adults who are socially isolated and lonely, they may report more health risk behaviors like smoking, alcohol abuse, physical inactivities, or disturbed sleep. And there is also psychological dysregulation pathways, although as Ted and Fereshteh just mentioned, it's a really understudied area, because there's only like inflammation pathways has been studied, and Ted and Christina has just recently started their investigation on this area. And also there's psychological distress pathway, which means social isolation and loneliness will lead to more depressive symptoms and anxiety, and therefore will lead to those health outcomes. But there comes a lot of questions. Why social isolation and loneliness are associated with so many diseases? As we delineate the three potential pathways, but
Think about it.
Whether it's a single organ mechanism or single system mechanism, or the aging process of human as a whole, and also are these health impact different between social isolation and loneliness? Yeah, that's why I look into whether accelerated biological aging could serve as a mediator. We know that aging is the greatest risk for diseases like cognitive impairment, cognitive decline, ADHD, cancer, arthritis. And also, if you compare aging with other well-established risk factor for disease, like alcohol abuse, like smoking, like obesity, we can see aging dominated the relative risk for any other risk factors on cancers, on heart disease, on Alzheimer's disease, etc. And the biological aging is also the proposed mechanisms through which social determinants drive health disparities. Because following the generalized paradigm, most age-related diseases are caused by the shared biological mechanisms of aging. And also previous study has theorized that biological aging may serve as a potential mediator on the association of social factors with phenotypic and functional aging. but very few study has tested the pathway of social isolation and loneliness. Therefore, the aim is to examine the association of social isolation with motoric cognitive risk syndrome, and also investigate whether accelerated biological aging would mediate these associations. This is a direct acyclic graph for the causal diagram. The exposure is a social isolation or loneliness in 2012 or 2014, and the mediator is measured by accelerated biological aging markers in 2016, and the outcomes is measured ever since. The dataset we use is from Health and Retirement Study. Yeah, I think it's probably the most well established study in the United States and probably in the world. I would also express my thanks to Dr. Sonnega, who is also on this panel. Yeah, for your great effort and the team for making this such a great study. And yeah, you know, we're standing on the shoulder of giants to use these open data sets. And also HRS has a rich set of health measures, including biomeasures in the Venus blot study in 2016, and the psychosocial measures, very rich measures in the left behind the questionnaire. And the social isolation is measured by the Steptoe Social Isolation Index, which measured people's marital status, living arrangement, social contact with their children, family, and friends, and the participation in social activities. And loneliness is measured by the 3-item revised UCLA loneliness scale. They measure people's frequency of reporting lack of companionship, feel left out, feel isolated from others. And the biological aging is quantified by a 3 DNA methylation marker: phenol age, grim age, and donating pool. DNA methylation is an addition or removal of a methyl group from across numerous CPG sites involved in gene regulation and expression. And it's probably the most well-studied markers of biological age because it's high degree of accuracy in predicting the chronological age. And it is associated with numerous age-related disease, including diabetes, cancers, cardiometabolic disease, etc. And the outcome is a motoric cognitive risk syndrome. It is a pre-dementia syndrome and characterized by subjective cognitive impairment and a slow gait, but in the absence of dementia and mobility impairment. The reason we chose this as an outcome is because it's a pre-dementia syndrome and it's most likely to be reversible. And if we can, you know, identify risk factors for this syndrome, we can probably prevent the effects of ADR, Alzheimer's disease, and related dementia. This is the sampling flow chart. Basically, we included people with valid variables on this measurement, but we excluded the people who already have the motoric cognitive risk syndrome as a baseline. And finally, we included like 2,000 people. And for the causal mediating analysis, we use data command made 4 ways, which is a 4-way decomposition of the total effects of exposure on outcome. It is based on the counterfactual framework and calculated the natural direct effect and the natural indirect effect and the proportion mediated, indicating the proportion of the total effects explained by natural indirect effects. And this is the sample characteristics according to categories of social isolation and loneliness. We categorize the people by their baseline exposure to social isolation and loneliness, and we estimated their biological age acceleration. We compared people who are isolated, not isolated, lonely or not lonely. We can see from here, for the phenol age marker, for those who are socially isolated, they are probably half a year accelerations in biological age. And for the grim age, also almost 1 year's accelerated biological age. But for the donating pool pace of aging, for those who are socially isolated, also have a accelerated pace of aging. But not for loneliness. For people who are not lonely or not lonely, they don't have any differences in the biological aging measures. And we first examined the association of social isolation loneliness and the onset of motoric risk syndrome, and its components like subjective cognitive impairment and the slow gait. When looking into the subjective cognitive, the model 1 is adjusted for social demographics. Model 2 further adjusted for health risk behavior, and model 3 adjusted for health outcome measures. When we look into its association with subjective cognitive impairment, we can see no matter how much we have controlled, social isolation has been associated with the risk of subjective cognitive impairment. And for the slow gait, when looking to slow gait, both social isolation and loneliness has been associated with slow gait speed. And for motoric cognitive risk syndrome, for loneliness, it is associated with this before controlling for health measures, but after we controlled for depressive symptom, BMI, comorbidities, the association disappeared. When looking to the mediating role of accelerated biological agent, we see here, Grimmage and donating poem mediated like 10% of the effects of social isolation on subjective cognitive complaints. And Grimmage and donating poem also mediated like 10% of the association between social isolation and slow gait speed. But loneliness is not. And for the motoric cognitive risk syndrome, it has mediated like 6 to 10% of the total effects of social isolation on motoric cognitive risk syndrome. Therefore, we concluded that social isolation was so significantly associated accelerated biological agent, high risk of subjective cognitive complaints, slow gait, and motoric cognitive risk syndrome. And also this study suggested that a biological agent is a potential mechanisms through which social isolation impact motoric cognitive risk syndrome. This study also has some limitations we need to acknowledge. First of all, although we have used the causal mediating analysis, but the causal inference also remains uncertain because of the observational nature of the study. And the DNA methylations of biological agent was only available in 2016. And we examined, because it is measured only in 2016, we examined the motoric cognitive risk syndrome from this year to maintain temporality for causal mediated analysis. But this short follow-up is kind of short that limited our implication. Therefore, there is a need for future studies with longer follow-up durations. For the implications, this study provided new knowledge by identifying social isolation and loneliness as a proxy determinants of deficits in healthy aging, and also contribute to developing a more robust evidence base for the pathways and different risk profile for accelerated aging. And most importantly, it provides a sensitive and a specific choice of biological measures that could be implemented to evaluate policy, program, and interventions. Thanks for your attention. This is my contact information. I welcome all kinds of inquiries and questions.
Thank you, Dr. Kai. It was a good presentation. Now the floor is open for any questions.
Let's take a look. Okay, I don't see any questions in the chat. Again, feel free to post your questions in the chat or just unmute and jump in. We have a nice group here today, small enough, I think, that we can have a sort of open discussion, almost as though we're in person together. So please just feel free to unmute, introduce yourself, and then pose any questions you have for our presenters.
I believe there's a question in the chat box from Shoin Kim. I want to learn more about the statistical models that all of you used for the analysis. How did you come up with the model, and what nature of the data fit to the model?
I can take that question.
Mm-hmm.
So it really depends on your research question. For example, in my study, I look at changes among variables. So when we want to look at changes, so latent growth models fit these kind of questions. So because you can figure out what is the relationship between the slopes of the variables and how changes among variables can impact changes in other variables like social isolation and cognitive function. So it really depends what is your research question. And then, for example, in bidirectional association or causality, is better to use like random intercept cross-like panel models, because this kind of model is really designed to look at the causality. And in terms of the large data set, yeah, sure. For example, for me, I use a structural equation model. So we really need large data set, like more than 600, or depends on your waves when you have more waves. So large data set fits better to the model. So I don't know, I answer your question.
That sounds great. Xiang, did you want to jump in as well? It sounds like.
Oh yeah, sure. Yeah, sure. Okay, based on I feel like Dr. Sonnega has a because she's in the social data platform, I think, right? She has been done great work on this, on compiling those all kind of large data sets. I feel like for US study, it has a large platform that compile like national health and aging interview surveys, health and retirement study, a lot of studies, etc. you can choose from. And basically just look into those literatures and how this data set has been used and the statistical models they have used.
Is there any follow-up, or Ted, did you want to weigh in on thoughts about that?
Yeah, I think pretty much both the panelists or presenters actually articulated the rationales and the response very well, because the question really dictates the model to be used. Of course, even the same question, you could use different models to actually answer slightly different sub-questions in the bigger question. So that really depends on the questions and the nuances the question has, because slight different nuances can actually utilize different models, which could answer slightly different questions, which eventually could lead to different insights and perhaps different papers.
Mm-hmm. I wanted to ask you guys if you wanted to comment or talk a little bit more about the role of genetics. HRS does have genetic information. I think when it sort of started and came in, there was if any of you have ever seen Jessica Fall present, she always presents this hype curve, right? It was like all this excitement about what genetics and telomeres were such a thing at one point, and it's like going to have, it ends up having not as much explanatory power, and then it almost just becomes de rigueur to include it. But I'm not sure. I wondered if any of you had any thoughts about sort of including these because there definitely are, certainly ApoE, and HRS releases a candidate gene file for cognition. But what's sort of the value, the role, the importance of including the cognition or the genetic information when you're looking at these models? And each or any of you.
Maybe I can start first. So
yeah,
in terms of genetic, of course, depending on, again, because GSA is multidisciplinary, so everybody comes from different disciplines. So to me, as a basic biology trained investigator, so who actually later on go and perform clinical and epi research. So of course, the genetic, like Amanda mentioned, it can be, well, it's very rather broadly divided into the more traditional genetic, which is the genotyping, and then there's the epigenetic, which is the more modifiable that can be influenced by social and psychological factors. So for the more ApoE is one example of the more traditional genetic factor that unfortunately one could not change unless technological advances. So for ApoE, I would think that, yeah, definitely that is a very good suggestions and pointers here for future research, because as far as the study that I have performed, we have not included ApoE as either an effect modifier or a covariate. So that definitely has impact on cognition, as we all are aware of. So definitely that is a very pertinent point for ApoE. Whereas for the epigenetic, that could potentially be one of the candidate mediators or pathway, because as mentioned earlier, epigenetics, I think that's also probably one of the main reasons HRS actually included epigenetic, because HRS actually focus a lot of psychosocial factors and how psychosocial factors can influence epigenetic, which is the so-called gene and environment interaction in a modified version. So that I think definitely could be one of the good candidate as well for gene expression, because a lot of biological markers that especially I presented just now, the 5 different biomarkers, they are a snapshot in a time. And as we all know, a lot of the markers, the 5 markers, HDL, LDL, especially HbA1c, they can fluctuate from sometimes even days to days. So whereas epigenetic might be more stable in some sense compared to these snapshot biomarkers, and definitely in a way more novel compared to the more canonical blood test biomarkers that everybody that have access to these blood tests. So yeah, I think that's a really great suggestion. Thanks, Amanda.
Thanks.
Yeah. I'm also considering expand my research by adding the genetic part. Yeah, but one concern, as I discussed with my PI, because there is only a subsample of the participants who has completed the genetic data collection. So yeah, because the PI want to expand the sample size, because after the link with those who has genetic components, sample size will significantly reduce. But I feel like it's a very worth try to include genetics, maybe ApoE. I saw, there's a already open data set for the genomics gene that's for the ApoE for alleles and already there. Yeah, I feel like it's easier to do for, especially for people who are not have biological trainings.
Yeah. So we have a really interesting, nice question in the chat. Could the speakers comment on clinical implications and the potential role of social prescribing, which I've never heard, but sounds very, very interesting. Any one of you, if you want to, it's a question for all 3 of you. So.
Yeah, I have a lot of thoughts on social prescription or prescribing. So that's another strand or area of my research actually. So yeah, loneliness, isolation, mechanism, and cognitive impairment, which we have talked about today, is my second line of research. So the social prescribing is actually more of my first line of research since 10 years ago. So yeah, maybe I can kind of comment a little bit on that, because social prescription or prescribing has been pretty so-called prominent, especially in the UK. As some of you might be aware that in the UK there's a ministry of loneliness, if I remember correctly. So so that has been very a lot of advocation for social prescription or prescribing actually overseas. And I think definitely that's something that is still so-called, I would say that they are prelim evidence and also definitely anecdotal evidence that it could work. But I think we require more robust RCT implementation effectiveness type of studies to actually so-called provide a firm conclusion before moving into the clinical side, because that's really the gist and also the spirit of evidence-based medicine, right? So without so-called the more translational and also more community validation of different types of social prescription, it would not be prudent to move them to the clinical setting as yet, I would say. And of course, a little bit of shameless plug. Yeah, I've done some related social prescription on mindfulness, gardening / horticulture therapy, and we have shown some prelim results on how this community-based social prescription can actually improve cognitive functioning, even in MCI, and psychological well-being, and potentially depressive symptoms, and also a wide range of biological markers that closely relates to inflammation. That would be the more prominent one. We have examined cortisol, BDNF, and a few other markers, but unfortunately, they did not seem to either be good candidate or response to those social prescription. So inflammation seems to be the more appropriate candidate markers for these kind of intervention, or they might be more responsive to this intervention. Because as I alluded to, and the panelists also alluded to, the stresses part is really the part that I think driving a lot of the mechanism of isolation and loneliness. So stresses is perceived by the brain and passed down to the stress pathway in the body, which further elicit inflammatory responses in the body. So that's the so-called chronic low-grade inflammation, or the people in the more basic science would call it inflammation and aging.
Thank you.
Yeah, I could also comment on that. Yeah, because my current project with the PI is to use a kind of social prescription way to improve the health outcomes of the community dwelling older adults, caregivers for people with dementia. Actually, I found that it is not an easy job, especially when conducting this in the US. It's hard to engage people who are already socially isolated and lonely. They're already maybe they're self-chosen, and it's hard to intervene on their social network because they are already childlessness, and they don't like to they live in a not very crowded neighborhood, a very quiet neighborhood. It's very hard to intervene on that. But I saw quite a lot of studies conducted in UK or in Australia or in Singapore, right? But very few study has been done in the US. There's also kind of barriers like race, ethnicity issue, like living desegregation, and also their gender issue, a lot of things. Mm-hmm. So, but it's worth a try. I see NIH has been allocate a lot of fundings on this area, but I think it's a very promising area in the future.
That's really exciting. Fereshteh, did you have anything you wanted to add? Or
yeah, I just want to add that it's really important to identify those who are isolated or lonely. So because most of the program, like age-friendly cities or mindfulness program, or those intervention are for regular people. So it's really important to identify who are socially isolated or who are lonely and frail, because those people mostly don't get involved in any kind of intervention. So right. I think that is the first step. And then we can have some social prescribing also for these kind of people that really leave behind.
Yeah, that's a really, really interesting question. And we're coming to the end of the hour, and I believe we'll go ahead and leave it with the last question is actually sort of a little bit of a technical one for you, Xiang. That 23% of the people were included in the venous blood size. You have this severe sort of sample selection that happens at that point. And they're wondering, sort of what this means, in terms of, this becomes a high sort of selected sample. Now, in theory, the way that the way that the sample was drawn in the first place is still meant to be reasonably representative, but you do have to agree to participate in the venous blood sample. So, but I think there's a fairly high agreement rate, so it's not as bad as you might think. But you're more expert than I am, so let me let you take that, and then I believe we'll be at the end of the hour. And thank you all. If we get cut off, thank you all for your answers, your great presentations, and for everybody who attended for your participation. So, Xiang, you're going to take us out.
Yeah, thanks for the great question. Yeah, actually, yes, it is. There's only 23% of people who are included in the venous blood study. Yeah, but actually, it's it has to came up with a compromise solution because, yeah, there's only this kind of people who respond to this question. You have to acknowledge it into the limitation part, saying that they're high selective sample. But another way to address this concern is to compare those who had dropped out, who didn't answer this venous blood study, and with those who had included in the study, and compare their characteristics. But actually, from what I've found in previous studies, is that there is not very much different characteristics when compared those who are included or not included. Just the only one criteria is that females are more likely to participate in those kind of venous blood studies or other kind of sub-study experimental modules. But other characteristics, they are pretty similar.
Yeah, that sounds right to me as well. You're getting lots of nice thank yous, and yeah, and so I think we're at the top of the hour, and I'll join all of your participants here today in thanking you, all of our presenters, for a wonderful panel. Really, really interesting. And again, this will be recorded and available, on the GSA website going forward. So I think that's it. Thanks again, everyone.
Thank you all.
Thank you.
Thank you.
Thank you.
Bye-bye now.
Bye.
Yeah, bye-bye.